So I'll stop there. Thanks.
Thank you, Peter. That was really exciting. I mean, to think that we may actually see real DNA therapy in our day is like amazing. And congratulations on this seminal
Thanks.
work. So, questions from anybody?
Yeah. I lost what? So the
question
biggest
is can you insert something larger than a point mutation?
we've inserted up to about 10 base pairs, but we haven't gone beyond that. So it's possible we might be able to put a LOXP site in, but I think it's less efficient at putting in big pieces. I think that's where CRISPR would have a big advantage.
Any
Okay.
other questions? Okay. Thank you, Dr. Glazer. So I'd like to also introduce now Joanne Sweezy, who I had the pleasure to work with as the Associate Director for Basic Science in the Cancer Center. She's the Ensign Professor of Therapeutic Radiology and Professor of Genetics, as well as Vice Chair of Basic Research. And she's going to talk to us about DNA repair and cancer. Thank you, Joanne.
Thanks. So today, this is hard to speak into the microphone and look this way. Okay, today I'm going to talk specifically about precision medicine from a DNA repair perspective. And my laboratory for several years has been working on DNA repair variants that have been identified in the germline through the massive next-gen sequencing efforts, and also DNA repair variants in tumors. And some of these we've actually identified ourselves here at Yale, and one of them is a RAD51 breast cancer-associated variant that I'm going to talk about today. So I'm going to talk about the case. This was a 38-year-old African-American female with a 2.1 centimeter infiltrating ductal carcinoma. It was ER/PR+, stage 2B. 1 out of 15 nodes were positive. And we identified a somatic mutation in RAD51. It's heterozygous, and it alters amino acid glycine 151 to aspartic acid, G151D. It's heterozygous, so we can also identify the wild-type allele in this tumor. This is just a cartoon that I redrew from CBio, and this is not precise in any way. I just wanted to give you two take-home messages. The first is that if you look in the TCGA, the Cancer Atlas in CBio, there are somatic mutations in the RAD51 gene. They're rare, but they're there. There aren't many hotspots at all. Actually, there aren't any hotspots unless you like to call 2 mutations a hotspot. And the other take-home message from this is that RAD51 G151D, the variant that we identified here at Yale, is not present in the Cancer Genome Atlas. So it's a new variant. So what does RAD51 do? It forms filaments on single-stranded DNA, and it functions in double-strand break repair. And I feel really a little overwhelmed by talking about double-strand break repair because my colleagues are the major movers and shakers in this field, Ryan Jensen, Patrick Sung, et cetera, in our department. But I'm going to talk about it anyway. So we have a double-strand break, and how are double-strand breaks formed? Mainly through ionizing radiation and other kinds of cancer therapies. So the double-strand break is recognized by the MRE11 RAD50 NBS1 complex, and resection occurs to leave 3' tails. RPA, single-strand binding protein, binds to those tails, and what happens then is that BRCA2, a mediator, escorts RAD51 and helps it to load onto those filaments. And people from Yale have played a major, major role in figuring this all out, Patrick Sung and Ryan Jensen, per se. Once RAD51 loads on to the 3' DNA and forms this filament, it catalyzes invasion of another strand. Here we're talking, really looking at catalyzing invasion of a sister chromatid. And you can see it forms a D loop. What happens then is DNA synthesis takes place, And we see formation of double Holiday junctions, named after Robin Holiday. Depending on which way they're cut, we can get non-crossovers or crossovers. And most of you probably had this in a basic genetics class. But I just wanted to be sure that everyone knew what Rad51 does. So this is an EM structure of a Rad51 filament. Each of the different colors are monomers of Rad51. And this is where the DNA is, in this fine red line. If we look at it from a different view, we're kind of looking down on it now. The DNA is in the center of the filament. The filament is actually there to sort of stretch the DNA, to change the pitch of the DNA, so that it can recognize homology in the other strand. G151 is not anywhere near the DNA. So we've positioned G151, we've just drawn it here in these black balls to show where it is. And then if we look down on the structure, you can see that it's way on the outside of the filament, with the DNA being in the center. So this does not interact with the DNA in any way. All right, so let's go back to the patient and we'll find out how she was treated. First, there was a resection. Post-op treatment included adjuvant ionizing radiation, Cytoxin 5-fluorouracil methotrexate, 6 cycles of that. Metastasis took place 13 months post-diagnosis to a lot of sites. She progressed on tamoxifen, doxorubicin, vinblastine, and mitomycin C. So the first question we asked was, is it possible that this one variant in the tumor conferred resistance to therapy, had anything to do with conferring resistance to therapy? So what we did was we decided to use MCF7 breast cancer cells, which are ERPR+, and we expressed this variant in these cells. We did not overexpress it. We're very careful about monitoring expression levels and choosing clones that express the exogenous protein at the same levels of the endogenous protein. That brings me to another point, that these cells have wild-type Rad51, just like the tumor. So we're expressing equivalent amounts of the exogenous, either wild-type or Rad51, G151D, with the endogenous protein. And then we looked to see if this variant could confer resistance. So first we looked at ionizing radiation. Here we're showing dose and survival fraction. Here we're looking in black at the empty vector and just expression of wild type in these cells versus expression of G151D. And you can see that cells expressing G151D are resistant to ionizing radiation. Same for mitomycin C. Here's the dose of mitomycin C and surviving fraction. Cells are resistant to mitomycin C. And doxorubicin, cells expressing G151D, the variant, are resistant. So we would conclude that this variant has a lot to do with the tumor being resistant to therapies. We've placed this variant in other cell lines, and I'm not showing you that, but the variant confers resistance in the other cell lines as well. Okay, so the next question we had, since Rad51 functions in recombination, we wanted to know if this cancer variant was able to function in recombination, and for this we used the very famous Maria-Jason assay, where we have 2 incomplete bad copies of the GFP gene. One of them contains an ISC1 site. If we cut with ISC1, we can actually induce a double-strand break. And if there's recombination between the donor and the ISC1 recipient, GFP will be produced. And we can look at levels of GFP, which are reflective of the levels of recombination using flow cytometry. So here, what I'm showing you is not only does G151D produce a lot of GFP or induce a lot of recombination when it's expressed in these cells, we were totally surprised, but it induces a hyperrecombination phenotype. This variant has one of the highest levels of recombination ever seen with HDR variants. Okay, so Rad51 can promote exchanges with the sister chromatid, and I talked about that when I introduced the topic. But what we're really talking about here, just in this little cartoon, is the replication fork might be coming along, and there might be some damage there. Could be some oxidized base, maybe a break of some kind. And what Rad51 does is it can catalyze, as I've told you, it can bind to the strand and catalyze strand displacement here in the form of a D-loop. So one way of looking at that is to look at sister chromatid exchanges. You can see harlequin structures, and I'll show you that in just a second. So again, we looked in cells expressing wild type and in cells expressing G151D, and you can see some of these harlequin structures in these cells. And if I quantify this for you, here now we're looking at sister chromatidic changes per nucleus, 0 to 5, 6 to 10, 11 to 15, and so on, versus number of nuclei. You can see that when we express wild type in these MCF7 cells, we've also done this in non-transformed cells, you can see that when we express wild type, we have low levels of sister chromatidic changes, but they're not non-existent, they're there. But you can see there are significantly higher levels of sister chromatidic changes in cells expressing G151D. So these are very hyperrecombinant. This supports the idea that this is a hyperrecombinant phenotype. As an aside, we also show that chromosomal aberrations are present. And just to summarize this, again in red is G151D, the variant. Cells expressing G151D have increased levels of chromosomal fragments, fusions, and breaks. And that's interesting. We decided, because we see all of these aberrations, we decided to just kind of take a peek and wonder if G151D, besides playing a role in conferring resistance to chemotherapy, if it could also possibly be driving a carcinogenic phenotype. And so we just looked at an invasion assay in these tissue culture cells, and you can see that cells expressing G151D have really higher levels of invasion. So this variant could possibly be doing not only conferring resistance, but could also be driving cancer. Okay, so now I'd like to come back to this to remind you that one of the things that RAD51 does is it forms filaments on single-stranded DNA. And so if it's present on single-stranded DNA, we wondered if we could see RAD51 foci in the cells. And so we just looked spontaneously. This might be hard for you to see, but we're looking here at RAD51 foci in wild type and G151D cells. The DAPI is just going to light up the nucleus. But you can see that RAD51 foci are expressed mostly in G151D cells. And this is only spontaneously. We have not treated these cells with anything. And you can see that a higher percentage of nuclei that are expressing G151D have RAD51 foci. So these foci are forming spontaneously. This is highly unusual at these high levels. And when we treat with ionizing radiation, we expect RAD51 foci to form in wild-type expressing cells, and it does. But we see significantly higher levels. So let me start here. Here's just ionizing radiation. 0 hours after recovery, there's not a big difference between the wild-type and variant-expressing cells. We wouldn't expect that because now we've put double-strand breaks into the cells. So we should see RAD51 in the wild-type cells. But as the cells recover from ionizing radiation, at 4 hours and 8 hours, we see massive numbers of RAD51 foci in these cells. So G151D is really very active. It's doing something very interesting. And it's repairing, I'll show you now, it's repairing double-strand breaks faster at a higher rate in cells cells expressing G151D versus wild-type cells. And so Peter mentioned gamma H2AX as a marker for double-strand breaks. So we looked at the gamma H2AX marker, and what we wanted to do was to look again at recovery to see if we could see gamma H2AX disappear as a marker for disappearance of double-strand breaks, repair. Because remember, if cells repair DNA damage, then they're going to be resistant to cancer therapy if they repair it better, right? So that's the problem here. So here we're looking at hours after exposure and the percent of nuclei with greater than 10. This is a really high standard, gamma H2AX foci. And so you can see we have the opposite picture here with these cells as we did for a focus formation, and that is that we see breaks are repaired faster. The gamma H2AX foci are going down faster in cells expressing this RAD51 cancer-associated variant. So then we decided, instead of just looking at a marker, we decided to really measure double-strand breaks using the neutral comet assay. And here, if there are breaks in the DNA, you can see these tails or these halos coming out of the nucleus. And you can measure the length of these. It's called the tail moment. And that tail moment is absolutely reflective of the levels of double-strand breaks in the cells. And so you can see, again, after we treat with ionizing radiation, we have double-strand breaks in the cells, and so you don't see much of a difference between wild type and the variant. But 4 hours and 8 hours after recovery, there's a huge difference in the levels of double-strand breaks in these cells. with very few double-strand breaks in the cells containing the cancer variant. So this cancer variant is rapidly repairing double-strand breaks, which is why tumor cells or any cell expressing it are going to be resistant to therapy. So again, here I want to remind you that the interaction with DNA is not altered. So G151D is on the outside of the filament. It doesn't interact with DNA. But because it's on the outside of the filament, we thought that perhaps it might interact more tightly or less tightly or whatever with other proteins that catalyze homologous recombination. And Ryan Jensen probed that, and we didn't find that to be the case at all. Instead, Ryan, who's critical to this project, because he's purified all these proteins, he's got all these great assays to look at recombination in vitro, performed a strand exchange assay. Well, he performed a lot of assays. I'm just showing a few of them. So the substrates are here. So he gives, in the assay, this is the DNA substrate. It has this nice tail that Rad51 can form a filament on. But he adds RPA to bind to it because that's what happens in vivo. And RPA presents a huge block to Rad51, which is why you need mediators to load Rad51 onto the DNA. But you can see he's done this assay in the absence of mediators. He adds Rad51, and he adds this double-stranded DNA, where now we're looking for strand exchange. We're looking for Rad51 to catalyze strand exchange, to take this piece, this red strand, and exchange it into this original substrate. And he can look at that by gel electrophoresis. You can take my word for it that this is a double-stranded DNA, and this is the product. So you can see as he increases the concentration of our RPA, you can see that the product becomes less and less. In other words, wild-type Rad51 cannot catalyze strand exchange in the presence of higher levels of RPA. But look at G151D. It just goes. It just catalyzes strand exchange at fairly high levels of RPA. And Ryan has quantified that for us here. And you can see at the highest levels of RPA, G151D protein is still catalyzing strand exchange. Okay, so again, this is the case. And what I want to remind you again is that this is a heterozygous mutation. We can see wild type and we can see the mutant in this tumor. And so the question then is what happens when Ryan puts both proteins into the strand exchange assay, when he puts G151D, the variant, and wild type. And what he decided to do was challenge them with a high concentration of RPA. And what's interesting is here at 20 nanomolar, if we look at strand exchange with just wild type alone, we would see barely anything. He adds just a tiny, tiny bit of G151D, and the strand exchange activity is massively increased. So in cells expressing both wild type and the cancer variant is going to help the wild type really catalyze strand exchange. Now, we have preliminary data suggesting that these can form mixed filaments on DNA, but that data is by no means conclusive at this point. But that's what we think is happening. So the cancer variant is dominant to the wild type variant. So these are various, in a paper published a few years ago, these are just various models of DNA damage tolerance, which is what we're talking about. We're talking about DNA damage tolerance, in other words, better repair in a sense. And here what we're really looking at, we think, is, again, and I diagrammed this earlier, a replication fork coming along, and there could be a break, maybe no damage, we don't know, maybe no damage at all, maybe an oxidized base, and there's a template switch and strand invasion into the other chromosome. And so if this is going on at the fork, that should mean that spontaneously the forks in Rad51 variant cells, the forks should be moving at a faster rate, and in the presence of damage, they should be moving at a faster rate. And so we decided to perform this replication fiber assay. So what do? We label the cells with IDU, we pulse them with IDU, and then we can damage them. In this case, we just treated them with HU or no treatment, and then we followed with a pulse of CIDU, and then we spread them by this experiment was done by gravity. You have to get the right size pipette tip box, and you have to get your slide just so. But now we've purchased a machine in therapeutic radiology to pull these fibers very nicely. So if anybody's interested, you can come and see me after the talk. This was by gravity flow. And you can see here are all the fibers. Here's untreated wild type and untreated G151D. And what we do is we measure the lengths of these fibers. And from the lengths, we can quantify tract length, which is directly proportional to velocity, replication fork velocity. So you can see in untreated cells, the forks are moving much more quickly in the variant cells, the variant expressing cells. And even in the